Okay, so I hope you had meaningful discussions. I hope you had meaningful and fruitful discussions, and that your issues have been clarified. So we now want to move on to the second part of our presentation. Remember, we're talking about health-based exposure limits, including validation and giving an overview and introduction. So on to part two now. So we just want to introduce the concept of health-based exposure limits. And when we left off from the last presentation, we said that the solution to the issues of health, the limit values being arbitrary, um, and also sometimes being overly costly, and sometimes not being based on any not being health based, was to then come up with the health based exposure limits. And this is something which has been used for over 50 years in the toxicological field, in an area of toxicology which is known as risk assessment. So we want to start by defining what a health-based exposure limit is. So a health-based exposure limit is a daily dose of a substance below which no adverse effects are anticipated by any route, even if exposure occurs for a lifetime. So I've highlighted some words there, and the words I've highlighted is daily dose. So it means when we're talking of a health-based exposure limit, we're saying that even if a patient is exposed each and every day to whatever dose we have calculated, we do not expect any adverse effects to occur. And this is even if exposure is to occur for a lifetime. And I think whilst I am here, it's important for me to then mention some of the words or terminology which is used. If you've been reading around this area, you might have come across what's called ADE versus what's called PDE. And in essence, ADE, which is the acceptable daily exposure, and PDE, which is the permitted daily exposure, are used interchangeably. And both of them refer to a substance-specific dose that is unlikely to cause an adverse effect if an individual is exposed at or below this dose every day for a lifetime. You find that PDE is a term which was used more in the guidances from the European Medicines Agency, and ADE has been used more from the guidances from ISPE. So they are used interchangeably. And at this stage, I should also hasten to say that PDE is an example of a health-based exposure limit. So PDE is contained in health-based exposure limits. So I hope the next slide will probably try to clarify them. So the derivation basically of health-based exposure limits has been there in toxicology for a long time. And like I said earlier, it falls under a subject known as toxicological risk assessment or risk assessment. And there are several health-based exposure limits which are set within the sphere of toxicology. For example, in environmental toxicology, you could have tolerable daily exposure. In occupational toxicology, which some of you might be aware of, you've got occupational exposure limits. In the food industry, they use more of the acceptable daily intake. And if you look at the drug development sphere, we talk about the first in human dose, all of which are examples of health-based exposure limits, which are limits that you basically are bringing in based on scientific data, that below these limits, we do not expect any adverse effects to actually occur. And ADE or PDE is a particular example of a health-based exposure limit. And we're saying if somebody is exposed for a lifetime, we do not expect any appreciable adverse effects to occur. But before we go on, we just want to take a detour and just talk a little bit about some of the basic fundamentals of toxicology in what I've referred to as Toxicology 101. Some important concepts to consider. I think I've got two major concepts here which will make us really understand or get a deeper understanding and appreciation of these health-based exposure limits. So the first concept comes from a gentleman called Paracelsus. We need to define what is a poison or what is a substance which has got the potential to cause harm. And Paracelsus came up with a very fascinating definition which anyone who has done toxicology should have come about. He said, all substances are poisons. This is paraphrased, by the way. There is none which is not a poison. The right dose differentiates a poison from a remedy. So he says all substances are poisons, but it's the dose which then differentiates whether a substance is going to cause any adverse effects or whether it's going to be useful. And this is particularly important, when we're talking about the pharmaceutical industry, because we're talking about medicines, isn't it? I mean, we all know that if you've got paracetamol and you take 2 tablets of paracetamol for a headache, what's that? Maybe 1 gram of paracetamol for a headache, you'll be fine and happy. The headache will go away. But on the other hand, if you were to take 50 or 100 tablets of paracetamol, not only will your headache go away, but you might actually also die in the process because of liver damage. So put simply, this concept by Paracelsus can be captured by this statement, that the dose makes the poison. And what it therefore means is that if you can actually reduce the dose of any substance and continue reducing it, you get to a point where we do not expect it to actually have any adverse effects. Although I said blanketly now, I mean, for those who've done toxicology, you know that if we're talking about, genotoxic carcinogens, this might not necessarily apply. But then for the purposes of our talk for today, we'll just stick to that the dose makes the poison. Now, the second concept is a concept which is embraced in both pharmacology and toxicology. And this is the dose-response relationship. So basically, looking at this graph which is on the left, and starting from the point with the 0, what we are saying is that as you increase the dose, and this is your x-axis, which starts with 5, 10, going up to 35. As you increase the dose, be it looking first at adverse effects, but also this applies for pharmacological effects, there is a point where we do not expect any effect at all. Why? Because the quantity is so small that even if there are any interactions with the receptors, there is no pharmacological action or toxicological action that occurs. And as we're moving to the right from 0, we go to 5, we see that the response, there is no toxicological effect occurring. It's still at 0 on the y-axis. We get to the first point where there is that circle and we've got our error bars over there. There's no response, and this is about 7.5, isn't it? Or 7, or maybe let's say 7 milligrams. There is no response at all that occurs. And when we move to 10 milligrams, there's still no response. So that dose, the highest dose, where there is no response or where there is no adverse effect, is referred to as the no observed adverse effect level. So our no observed adverse effect level in this graph is 10 milligrams. It's not 7 milligrams because that's not the highest. Yes, at 7 milligrams, we do not expect to see any adverse effects from that graph at all. But what we try to look at is the highest dose where we see no adverse effects, and this is called the no observed adverse effect level. So, in the perspective of toxicology, we would call it a no observed adverse effect level. But if our response of interest here was a pharmacological effect, it would then be called a no observed effect level. And these things, they become clearer as you start to read many papers and derive some of the health-based exposure limits for yourself. Then as we continue increasing the dose, so take this as an actual experiment where 1, 2, 3, 4, 5, 6 doses were given. And these are represented by the little circles that we have on the graph. So in this particular experiment, they gave a 7 milligram dose, and then they gave a 10 milligram dose. Then the next dose which they gave was approximately 18 milligrams. And at 80 milligrams, we see that there's a response which is occurring probably around, there's a 20% response or thereabout. Now, the lowest dose at which we see an adverse effect in a particular experiment is referred to as the lowest observed adverse effect dose, or level, otherwise known as LOEL. So LOEL is an actual dose which was used in an actual experiment. So we can't say a LOEL in this graph that we have is 15, because at 15 milligrams, there's no dose which was given to those animals. The animals were dosed at 7, at 10, at about 18, at around 22, probably around 27, and then the last dose was around 32. So the lowest dose where we did not expect an adverse effect is called the LOEL. And if this had been a pharmacological response, this dose would be called a LOEL, a lowest observed effect level. I hope that makes sense. Now, generally, when we do such experiments where we get the NOAEL and LOEL, these are ordinarily subacute, subchronic, and chronic experiments. And as a general rule, we do not use, LD50 values or values from acute studies to come up with NOAEL or LOEL values. And I hope as we continue speaking, you'll see the reason why. Because here we're talking about medicines. You remember our definition of health-based exposure limit talked about a lifetime. So we're not just looking at one, administration of the drug, but we're assuming that this patient will continue taking this drug over and over and over a period of time. So generally, when we're doing our calculations, we want data which is derived from repeated doses so that we can then try to extrapolate it into our own human setting using the various adjustment factors which we'll talk about later. I hope that makes sense. So the two concepts that we have, we said the dose makes the poison, and then the issue of health-based exposure limits. So just talking a little bit more about these health-based exposure limits. So these are the two. So the first one, the dose makes the poison on the left, and then this is the dose response curve that we were talking about. All right, so moving right on. So this graph is probably one of the most important ones, this figure, in terms of trying to explain the concept of health-based exposure limits. So we'll start the explanation from the right. And this is where we've got the orange curve. So let's assume that this orange curve represents a dose response curve for a certain toxicological response when you utilize a certain compound. So we can tell from this that at 185 milligrams per kg body weight, there is no response. And that 185 milligrams per kg body weight also happens to be the first dose that we used. So it therefore can be defined as our NOAEL. And remember from our earlier definition, the NOAEL is the highest dose below which you do not see any adverse effects. So this is the no observed adverse effect level over here. I guess before I move on, maybe I should just also make mention of what BMD is, which is the benchmark dose, which is something it's a dose which is computer generated, so to speak, with the use of some statistical methods. And it's ordinarily used when you do not have a NOAEL, but perhaps you've got a LOEL, and using the LOEL value, you can actually calculate backwards to then come up with the BMD or the benchmark dose. But anyway, let's work with the NOAEL for now. So at 185 milligrams per kilogram body weight, there is no response. But as you continue increasing the dose, moving to the right, we see that there's a response going up there. So when we are working with our health-based exposure limits, basically what we're trying to do is, because the first studies are ordinarily done in animals, which is what the orange line represents. But then we know that human beings are not animals. So by virtue of that, there is always some form of safety factor which is put in to account for that interspecies variability. So in this particular example, which is just a hypothetical one, an arbitrary number of 10 has been used. But you find that when you're actually doing your PDE calculations, this number for interspecies variability varies depending on the type of species that you're dealing with. And in this case, we've got a little Mickey Mouse. If it is a mouse, ordinarily would use a safety factor or a value of 12. So what we then say is that if the NOEL is at 185, just because human beings are not animals, to be on the safe side, what we'll do is to then divide that value of 185 by 10. And we are saying that we would expect that if a similar study was done in human beings, again, this is hypothetical, we would have the curve, that's the brownish or purple curve, which comes up there. But here, remember, what we're looking at is the NOEL, which means we're saying below this dose, we do not expect any adverse effects. And we put a safety factor of 10 to account for any interspecies differences. Because perhaps human beings might happen to be, a bit more sensitive to the compound, and therefore the value of 185 might be too high. But then if we put this safety factor by dividing this value by 10, we can then get up to 18.5, which is what we have there. But then now that we've got human beings, we're saying even amongst human beings, there are intraspecies differences or intra-individual differences. That if you look at a population of humans, there are some who might be ultra-sensitive and some who are less sensitive, some who are hypersensitive and some who are hyposensitive. So to account for those who might be extra sensitive to the compound, we can then use another safety factor, in which case we divided the value of 18.5 by 10 to then come up with 1.85. So the whole idea really of the health-based exposure limits or the risk assessments that we do in toxicology is to ensure that any adverse effects, the likelihood of any adverse effects occurring in a human being is very low, close to zero. And here I've said the aim is to ensure that the unintended recipient is exposed to a dose too small or small enough to avoid toxicity. So we're actually just trying to push the figures more to our left. So when we apply all those different safety factors or adjustment factors, the whole idea is to try and move that value from the right all the way to the left. And like I said here, this is done through the use of uncertainty or adjustment factors, which I shall talk a little bit more about. I hope that makes a bit of sense. I know, if it's the first time perhaps hearing it, if you do not have some background in toxicology, it might all seem a bit confusing. But as you continue to start doing examples, you'll find that it's not that difficult a concept to grasp. So having said that and having given the background, I just want to talk a little bit about the genesis and progress of health-based exposure limits. And basically this is a concept which seems to have been borrowed from the food industry or food toxicology or risk assessments for food substances. And the International Society for Pharmaceutical Engineering, or ISPE, in 2010 published their risk map, which is the risk-based manufacturing of pharmaceutical products. And this is where we start seeing the issue of health-based exposure limits starting to be put out by ISPE. There's a more recent edition of the risk map, which is the 2017 edition. And then around 2014, the European Medicines Agency put out some guidance in terms of the need for and the calculation of health-based exposure limits, specifically the permitted daily exposure. And ISPE, as I said earlier on, instead of the health-based exposure limit that they used is the accepted or acceptable daily exposure, which in the 2014 European Medicines Agency guidance is basically used interchangeably with the permitted daily exposure, or PDE. And then more recently, we see some guidance from the American Society for Testing and Materials, which was published 2019-2020. And then in 2020, the Pharmaceutical Inspection Convention also had their guidance, which is basically in line with the European Medicines Agency one. And then the WHO TSR 1033 Annex 2 also describes the procedures of how to calculate these health-based exposure limits, basically in line with the EMA guidance of 2014. So just in a few slides, I just want to quickly go through, the different calculations from these different organizations or organs. So the ISPE risk map took toxicological risk assessment approach, which was used in food additives. And like I said, the issue of risk assessment has been part of the toxicological field, or the field of toxicological sciences. So in food, they use what's called the acceptable daily intake, and the ISPE came up with the acceptable daily exposure limit, also the acceptable daily exposure limit. And again, just remind ourselves what the definition of ADE is. It's a dose that is unlikely to cause an adverse effect in an individual if an individual is exposed to it by any route at or below this dose every day for a lifetime. And that's basically what PDE is, isn't it? And in terms of the actual calculation, they took the NOEL. I hope you remember what NOEL is. So it's the NOEL, which is usually expressed in milligrams per kg body weight per day. And that is then multiplied by the body weight. I think the ISPE document, the body weight which they suggested was 60 kgs in an adult. You find that the EMA guidance, you use 50 kgs. And then this is divided by some composite uncertainty factor. And if you recall in the previous slide, the other slide with the graph with the mouse and the man, we had two uncertainty factors being put in there. One was for interspecies variability, which was a 10, and then the other one was for intraspecies variability, which was another 10. But then there are some other factors which can also be put into this. For example, if you have a LOEL instead of a NOEL, some of which we shall talk about in slides which are to follow. And then they also have what is called a modifying factor. Ordinarily, this is where your professional judgment will come in as a toxicologist or a person who is doing these calculations. And then they have some pharmacokinetic factor to account for the pharmacokinetics of the different compounds. So this was the way that the health-based exposure limit was suggested to be calculated under the ISPE risk map of 2010. And again, just to remind us that the whole idea of the health-based exposure limits is to try and minimize this dose, the NOEL that you get in an animal study, assuming that that's the data that you have, to try and make it make sense in a human beings, in a human population, by the use of adjustment factors. And then the second one that I want to talk about is the present one, which most regulatory authorities actually look at, which is the EMA or the WHO guidance. And in this, instead of using ADE that we saw before from the ISPE guidance, there is the use of PDE, which stands for permitted daily exposure. It's expressed in milligrams per kgs. And just like the formula that we saw from the ISPE one, you can either use a NOAEL or a LOEL value. Why LOEL? Well, sometimes it is possible that the first dose that you actually use in a toxicity study may actually have toxicological effects, and therefore in that study you will not be able to come up with a NOAEL value. So instead of saying, oh, this data is useless, let's throw it away, you can still use the data and then account for there being a lack of a NOAEL value by applying some adjustment factors. And we've got a body weight adjustment, which we saw again in the ISPE guidance. And all this is divided by various adjustment factors. The beauty about the EMA guidance or guidelines is that these adjustment factors are actually defined. For instance, the F1 would account for extrapolation between species, and that would have been the 10 that we saw in that the diagram that I had. And then another 10 for intra-species differences or variability between individuals, which was the other 10 that we saw in that graph. And then you could have some studies which are of varying duration. And remember, when we talk of ADE and PDE, we are talking of a lifetime. So if a study has been done in animals, perhaps over 1 week, that cannot be defined as a lifetime. So you have to now try and put in some other safety factors or adjustment factors, to take a more conservative approach, because you do not, truly know whether taking a substance for a very long time is going to increase the toxicity or not. So that's what F3 stands for, the duration of the study. And then we have got another one, which is F4, which is applied in cases of severe toxicity. And ordinarily, severe toxicity is defined as toxicity that might lead to death, perhaps teratogenicity, perhaps irreversible neurotoxicological effects, maybe carcinogenicity, as well as related very serious effects. So if in such cases you really want to make sure that the dose is reduced, because then that increases the safety, the conservative approach will at least give you a bit of a peace of mind that you are reducing the dose as much as possible to get close to the NOAEL as much as possible. And then there is another factor, which is F5, and this one is actually used when the point of departure, which is something that we shall talk about, I hope, maybe in another lecture, is not a NOAEL. Like if you look at that formula, you have NOAEL or LOEL. So if the value that you're using at the top is a LOEL value, then you have to account for it by giving it a certain value on F5. So basically, I think this is the formula which is currently being used and is probably the most important one at the moment. And then the ASTM E3219 formula is more or less really the same as the EMA one. If you see instead, so they've got their point of departure right there at the top, which can be NOAEL, LOEL, benchmark dose multiplied by the body weight. We saw this in the other formula from EMA. And then this is divided by the denominator. They've FT, which is the composite adjustment factor. So this would be your F1 times F2 times F3 times F4 times F5 in the previous slide. But then they have what they call PKAF, which is accumulation factor to account for compounds which actually accumulate in the body. And then they have another one, alpha, which is their bioavailability for the route of exposure. But really, more or less, whether you go using this route or you use the previous one, with proper judgment and experience, you come up with more or less the same type of answers. And then just quickly, this is a slide which is showing the different comparisons between the amongst the different organs. That's EMA, WHO, ISPE, and ASTM. So yeah, I think for this part, this is where I would like us to end. It's been about 30 minutes of me talking. So I know if you've not come across this, it might appear as though it's something really remote and out there. So I'll take this opportunity to entertain some questions. And then let's go into our groups and just discuss concerning this part of the lecture. And then we'll meet very soon for the third and final part of this particular talk. So until we meet again, just go into your groups and just start discussing.